Hi,
Hi.
I'm Sadashiva Pai from Science Mission. Today we have Dr. Rochelle Hines from Universities of Nevada with us. She has won several awards. In 2007, she won the Senior Graduate Fellowship Studentship from Michael Smith Foundation for Health Research. In 2009, she won a postdoctoral fellowship from Canadian Institute of Health Research. In 2011, she won the Pilot Project Award, Center for Neuroscience Research Tufts University. In 2017, she won Faculty Research Mentor Award from the Office of Undergraduate Research at UNLV. So welcome, Dr. Hines.
Thank you very much.
So can you please talk about your background, interest, and some research aspect of it?
Yeah, sure. So my background is in neuroscience. I got my bachelor's degree in neuroscience as well as my PhD, and then did a postdoc after that at Tufts University in Boston. My PhD and my bachelor's degree were earned in Canada. I'm originally from Canada, so you'll probably notice my accent as we talk today.
Yeah, a little bit, yeah. And tell us about your lab and grad students, postdocs, just briefly.
Yeah, I'm really lucky to have a great collaborator who also happens to be my husband. So we actually run our labs as a group collective. And between the two labs, we have 6 graduate students and a number of undergraduate students. We don't yet have senior people postdocs, but we're hoping that that's right around the corner.
Okay. And how do you motivate these students?
I think a good motivator is leading by example. Try to show an example of hard work. And it's hard for me to contain my enthusiasm sometimes for projects. So I think that can be a bit contagious too. I'm always really excited about what we're working on and moving things forward and trying to get to the next steps. So.
Okay. Can you please talk about your role models, mentors in your career?
Yeah, I've had a lot of really great mentors. So the labs that I worked in through my training, Dr. Jeff Kleim, Dr. Ala El Husseini, and Dr. Stephen Moss have all been really influential. I think Ala is particularly influential for me because while I was a graduate student, he passed away, and so that was kind of a really polarizing event. And I often kind of think of, you know, what advice he would be giving me if he were still here. And I'm also really motivated and guided by women in science. It's kind of a passion of mine, successful women in science. And so I look to a lot of the kind of, you know, more classic role models like Brenda Milner and Rita Levi-Montalcini, but also look to a lot of my contemporaries who are, you know, doing really incredible work, really advancing neuroscience research.
Okay. So let's just briefly talk about what else our interests are other than the research, like hobbies, and I know you have a couple of small children.
Yeah. Well, I grew up in Canada, right next to the Canadian Rockies, so I'm kind of an outdoors person. We do a lot of hiking, backpacking, rock climbing, and Las Vegas is a great spot for that because there's a lot of, you know, parks within a day's drive. So we do a lot of outdoor trips.
Okay. And coming back to your research, what made you get interested in neurodevelopment?
So I actually started my undergraduate degree thinking about heading into special education. And at the time I was working in schools with kids with autism. And I kind of became frustrated because I felt like we didn't even really understand the fundamentals about what was happening for these kids. You know, we didn't really understand what was going on inside their brains. We didn't really understand the pathophysiology. And so I got really interested in kind of trying to learn more. And then I just kind of fell in love with neuroscience.
Okay. So how does your current work bridges to whatever your instincts? Can you please go ahead and present your work whenever you get a chance?
Okay. Yeah. So today I'm going to be talking about research focusing on GABAergic synapses. And let's see. I'm forgetting. And I really got interested in GABAergic synapses, seeing the powerful role that GABA plays in development. And so that's been kind of an ongoing topic for me, is looking at inhibitory synapse development and how this contributes to some of the pathologies like neurodevelopmental disorders. And we'll touch on some of that today.
Okay.
So the title for my talk is GABAergic signaling at the axon initial segment. You can see the slides well?
Yeah.
Okay, perfect. So first, I'll kind of talk about why brain development is actually such a complex task. We know that disorders of the brain of brain development are actually quite prevalent. There's more than 4.6 million children affected in the United States. And they're quite heterogeneous. There's a number of, subtly different disorders that kind of get grouped together. And also many of them are polygenic. So there's autism, for example, has about 65 genes that are strongly linked, and more than 200 others that have weaker links. So it is quite complex in terms of, how these disorders come to be. But we can actually gain quite a bit of benefit by looking at what's common among them. And so we do see that there's common symptom profiles. So things like impaired social skills, problems with communication, repetitive behaviors, and also things like sensory hyperarousal and epilepsy. And epilepsy is particularly interesting to me. It's comorbid across a number of different neurodevelopmental disorders. And we also know that there are some overlapping genetic contributors. And what we see is that a number of genes involved in synapse formation and control of the formation of neuronal circuitry actually are represented across multiple neurodevelopmental disorders. So we do actually understand a great deal about how to set up the cortex, for example. And, a lot of really elegant work over many years of research has shown the steps involved in, developing new cells and having them migrate up the radial glial processes, sending out their own processes, and developing synapses. And although we understand all of this kind of very well as a model, the trouble with building a brain is that it doesn't really look like this. It actually looks a lot more like this. And what we can see here in this really beautiful image from the Brainbow 2 project is that it's really, really complex. The cells have complex morphologies. They have a number of interacting partners, and those partners need to be very highly specified. And in this image, we're just seeing just a tiny portion of the cortex, and not even, we're just seeing only neuronal cells. We're not even seeing the non-neuronal cells here. So this gives you kind of a glimpse into why it is so hard to set this up perfectly every time. And if we kind of step back out to a model level, we can actually see that there's a lot of different types of cells that make up the cortical circuitry, and they all have their own kind of layers in which they reside. They have unique morphologies, and they have very specific patterns of connectivity. And it's really important that we have this high fidelity wiring during development in order to have normal cortical function. So in typical development, we virtually never see a mistake. The right synapses always form on the right targets, and everything proceeds as normal. But subtle modifications to this can lead to disordered states, like disorders of development. And when we do have appropriate wiring, we get the generation of patterned activity. And so, everybody's heard the term brainwaves. That's something that I'm really interested in my research, are these patterns of activity, these brainwaves that we can record using electroencephalography. And underlying normal states, we see very kind of characteristic patterns. And if we kind of disrupt the pattern moving into epilepsy, we can see that activity runs out of control, and we see high amplitude and repetitive firing of cells, and this leads to visible seizures. And then kind of on the other end of the spectrum, we can see kind of slowing of the oscillatory activity underlying states like sleep and anesthesia. And eventually, if we slow it down enough, we can see highly pathological states like coma. And so the way that we kind of pattern activity is really important for supporting different functional states and different states of arousal. And we know that the peaks in the EEG are characterized by on signals or excitation. And so this peak here would represent a large population of neuronal cells firing in relative unison. And where we see these troughs in the EEG, these are characterized by off signals or inhibition. And if we didn't have inhibition, we wouldn't see this kind of shaping of the activity. We wouldn't see this development of patterning. And so, again, many years of research has kind of demonstrated that patterning of activity is really powerfully shaped by synaptic inhibition. And the primary inhibitory neurotransmitter in the brain is GABA. And so here's a look at an inhibitory synapse in an electron micrograph. And what you can see, all these little circles here are the presynaptic vesicles. They contain GABA that's going to be released onto this postsynaptic compartment here, which in this case actually happens to be a shaft of a dendritic spine. Or a shaft of a dendrite, sorry, not a spine. And here we're looking at a cartoon of this kind of same sort of inhibitory synapse. So here we have the presynaptic terminal with these vesicles filled with GABA. And then GABA is released into the cleft and it crosses and binds to postsynaptic receptors that we're going to talk about in a few minutes. GABA is really fascinating to me because it does play some key roles early in development. It's actually the first neurotransmitter during development, and it plays roles in guiding cells to the appropriate layer and also in promoting their maturation. And so in terms of trying to understand neurodevelopment, GABA seems to be a really good place to start, given that it plays all of these really critical roles. GABA also controls these unique developmental oscillations called giant depolarizing potentials. And that's because early in development, GABA is actually depolarizing because the chloride gradient is not yet set up to be high outside of neuronal cells. And then as GABA becomes hyperpolarizing, as it looks in the mature nervous system, we see that these giant depolarizing potentials disappear. But this also creates a seizure-sensitive context. And we can see that there's a very high incidence of seizures in development and also in neurodevelopmental disorders. So here we're looking at the incidence per every 100,000. You can see that it's highest in the first 10 years of life and goes down. And then later in life, it goes back up again for a number of reasons. But we're just kind of focusing on this area here where very early in life, we see this relatively high incidence of seizure disorders. And then if we look at the intersection between neurodevelopmental disorders like autism, Rett syndrome, Angelman syndrome, we can see a lot of things in common, including a relatively high incidence of seizures in these disorders. So now we'll talk a little bit about the postsynaptic target of GABA, because the postsynaptic compartment is one way to possibly modulate GABAergic signaling. So again, when GABA is released from the presynaptic terminal, it crosses the cleft and binds to postsynaptic receptors. And the most common postsynaptic receptor for GABA is the GABA-A receptor, and it's a ligand-gated chloride channel. And again, because the chloride is normally high outside of neuronal cells in maturity, opening the GABA receptor channel most commonly results in hyperpolarization of the postsynaptic cells. So chloride will move in and bring negative charge with it. GABA-A receptors are also super interesting because they're the site of action for a number of clinically important drugs like benzodiazepines, barbiturates, general anesthetics, and also sites of action for alcohol and many neurosteroids. GABA-A receptors are super heterogeneous. They're heteropentamers, and they're composed of 19 possible subunits. The most common GABA-A receptors contain 2 alphas 2 betas, and a gamma subunit. And again, over a number of years of research, we've really learned that the subunit composition influences both the function and the localization of these receptors. So in terms of function, we can see differences in the sensitivity to GABA and other ligands, and also in things like decay time. And in terms of localization, we can see that these different types of receptor compositions actually end up at different types of synapses. So there's also diversity in the subtype of inhibitory synapses. And we see actually a kind of incredible diversity in the different types of interneurons, and they all have their own pattern of connectivity. So each of these colored cells is a different type of GABA releasing cell called an interneuron. And when these cells release GABA, they have a number of different effects on the postsynaptic cell. So here we can see a cell that targets the dendrites. And because of the decremental nature of graded potentials, these have a relatively local effect. But we can also see synapses here on the soma, and some that occur right on the axon initial segment. And because these are very close to or at the site of action potential generation, we they kind of exert a lot more powerful control over the patterning of activity from these pyramidal cells. And these inhibitory synapses right on the axon initial segment have kind of captured my attention for a long time. I first got interested in them as an undergraduate because I was looking a lot at electron microscopy, and I was fascinated by these very unique synapses that were coming onto the axon initial segment. And here's just a really beautiful picture of this specialized type of interneuron called the chandelier cell. And it's colored in red here, and it sends out this axonal projection that branches and terminates on the axon initial segment of large populations of pyramidal cells. So this one chandelier cell is potentially signaling to huge groups of pyramidal cells all at once, acting to hyperpolarize or inhibit them. And in this way, we could see that this cell type in particular might have really powerful control over the phasing and the coordination of activity of cortical cells. And here we're looking again at an electron micrograph. This is much like the images that kind of captivated me as a student. Here's the cell body coming down here in the axon, and you can see a number of inhibitory synapses occurring right onto the axon initial segment here. So some of the overarching questions of my work are, how are these functionally diverse GABAergic synapses specified during development? What role might they play in neurodevelopmental disorders? And specifically, I'm interested in how GABAergic synapses on the AIS develop, what role GABAergic AIS signaling plays in regulating developmental activity patterns, and again, kind of the functional implications for any dysregulation that we might see. And so really what we set out to do in these experiments is to perturb GABAergic signaling at the axon initial segment. So the first thing that we started working on is what is unique about alpha-2, and how do alpha-2 containing receptors end up getting enriched at the axon initial segment? Because they do seem to have this preferential localization there. And so here's a look at a sequence comparison between alpha-1 on top and alpha-2 on bottom. And every time there's a red dash here, you can see that means it's identical between the two subunits. And hopefully you can see there's a lot of red dashes, except in this one segment here. And this actually represents a portion known as the large intracellular loop, and it lies between transmembrane domains 3 and 4. And so because alpha-2 ends up at the axon initial segment preferentially, and alpha-1 ends up there with a much lower frequency, we became interested as to whether this portion may be responsible for that preferential targeting. And in collaboration with Hans Merrick and Hermann Schindlin, we were able to demonstrate that there's a specific interaction for the alpha-2 subunit with collybistin. And for a long time, it was thought that Geoffrin was the preferential interacting partner with alphas 1 through 3. But again, we see kind of a reciprocal preference for interaction between alpha-2 and collybistin, and then Geoffrin is a preferential interactor for alphas 1 and 3. And so that's included in our recent paper. They did a lot of really beautiful work to demonstrate the strength of this interaction. So once we knew that that portion was specific for interacting with collybistin, we wanted to kind of see if we could disrupt that interaction and also disrupt the axon initial segment localization. So we generated an alpha-2, alpha-1 chimera, and we were able to show that it does not interact with collybistin here using immunoprecipitation. And we were also able to show in a separate set of studies that these chimeras don't actually reach the axon initial segment at the same rate. So here's an image of the axon initial segment, and these are clusters of the normal alpha-2 loop. And then here's when we substitute this portion with alpha-1, we can see a dramatic reduction in the clusters on the axon initial segment. So next we set out to really investigate what functional impact losing alpha-2 containing receptors might have on the AIS and on just kind of function in general. In order to do that, we generated a mouse model. And one of the first questions that we wanted to investigate is whether GABAergic synapses would still be present on the axon initial segment, even in the absence of alpha-2 containing GABA receptors. So here's a look at the mouse that we generated. So wild-type mice will have a normal alpha-2 subunit. And then the mice we generated, we called Gabra 2-1. And they have a small substitution in the large intracellular loop. This is about 13 amino acids from alpha-1 that's been substituted into alpha-2. So everything else about the subunit looks identical except for these 13 amino acids. We can test for the validity of our model using a loop antibody. So this antibody is specific for the alpha-2 loop. And we can see in heterozygous animals, the band for the loop is reduced. And in homozygous animals, it's almost completely absent. But interestingly enough, when we use an antibody against the C-terminus of alpha-2, we see actually that total expression of alpha-2 is increased. So again, heterozygous animals have a mild increase in alpha-2 expression, and homozygous animals have a relatively strong increase in alpha-2 expression. And down here, we started to look at some of the other interesting targets, including Geoffrin and collybistin. And what we found is that Geoffrin remains relatively unchanged. And again, this is in agreement with the idea that Geoffrin is probably not interacting as strongly with alpha-2 in comparison to alphas 1 and 3, whereas collybistin would be the preferential partner. And again, in heterozygous and homozygous animals, we see a significant decrease in the expression of collybistin. In collaboration with Jean-Marc Fritschie's lab, we also did a lot of immunohistochemistry to look at a number of different subunits as well as a number of different inhibitory proteins, but I'm just going to highlight a couple here. So here's confirmation that the total alpha-2 expression is increased. So normally in wild-type animals, we have, a kind of moderate level of puncta. And in both heterozygous and homozygous animals, we can see an increase in the density of puncta. But we do see a decrease in size. And we also stained for collybistin, and we're able to see that in agreement with the Western blotting data, we see a reduction in both heterozygous and homozygous animals in terms of collybistin cluster size and collybistin cluster density. We did a lot of investigation about the total number of inhibitory synapses, and we found that the total number of VGAP puncta wasn't changing. But we wanted to see if there might be a specific loss on the axon initial segment. And so we started out in neurons that were cultured from either wild-type or homozygous animals. And what we found is that in wild-type animals, we could see clusters of alpha-2 on the AIS. But when we looked at the Gabra 2-1 homozygous cultures, we saw a loss of those alpha-2 containing clusters. And we've been able to validate this in the animal model as well. So here we're looking at an axon initial segment stained with beta 4 spectrin in green. And we're looking at VGAP positive inhibitory synapses on that axon initial segment in red. And you can see that the inhibitory synapses kind of occur throughout the length of the beta-4 spectrum in wild-type animals, but we really don't see that accumulation in the mutant animals. So next we wanted to see what influence this loss of synapses onto the AS might have on postnatal development. And one thing that we actually noticed right away is that a proportion of the Gabra 2-1 pups were dying during postnatal development. And so we started kind of plotting and tracking, how many pups were dying and at what age and of what genotype. And so here we're looking at the proportion found dead across the lifespan between 0 and 120 days of age. You can see that very few animals die in the first 5 to 10 days of life. And we see a peak of mortality somewhere around postnatal day 20. And then it tapers off again and stays relatively low throughout the rest of the lifespan. And we can see a similar thing here looking at this survival plot from heterozygous by homozygous mating pairs. So each litter starts at 100%, and then as animals die, the line drops down, and we can see that, again, we have this clustering of animals dying somewhere around the age of postnatal day 20. We also analyzed the genotypes of animals that were dying, and we found no wild-type animals were dying. So that was reassuring that we were looking at something specific to the mutation. And we also found that looking at cumulative mortality over time, both heterozygous and homozygous animals have the same cumulative mortality, but homozygous animals seem to kind of die earlier in development, with heterozygous animals being pretty fixed right around postnatal day 20. And what we also started observing during that period of time was the incidence of spontaneous seizures. And so here's looking at Racine scores for a handful of the spontaneous seizures that we observed. And so a Racine score is just a behavioral method of scoring seizures. We were able to see that many of the seizures that these animals were experiencing were of highest severity, and they were happening right in the age range that the animals were starting to appear dead. So we also got interested in what influence axon initial synapse loss would have in mature animals, because interestingly, not all of the animals die. And in fact, a relatively small number of animals are actually able to be observed having seizures. So we wanted to see in those animals that do survive, what's going on in terms of their function. And so we had a close look at the surviving Gabra 2-1 mice, and we found that they had normal growth. So they weren't subject to some sort of failure to thrive or other, gross developmental deficit. And we also did a Sherpa screen. And so this is just a basic screen of sensory motor and reflexive function, overall health. And we found that the heterozygous and homozygous animals did not significantly differ from wild-type animals on the total Sherpa score. And we also put the animals through a number of tests like the rotarod to examine their motor function. And we also put them in the open field. And that's the data shown down here. This is looking at the cumulative distance traveled while the animals are in the open field, and also the average speed at which they travel while they're in the open field. And we didn't see any differences there. So this gave us some confidence that we could possibly examine other phenotypes that might be of interest in the mature animals. Alpha-2 is also highly implicated in anxiety, and alpha-2 containing receptors are thought to be a primary mediator of the anxiolytic effects of benzodiazepines. And so anxiety was kind of tops on our list for something to examine. We used two different tests of anxiety. We used the light-dark boxes and the elevated plus maze test. And the premise here is that rodents will prefer to be in a relatively safe, enclosed, dark compartment in comparison to a brightly lit, exposed compartment. And again, the premise is similar here, where we assume that rodents will want to spend time in these enclosed arms where they feel safer, as opposed to venturing out on these open arms. And so in each of these tasks, we're looking at the amount of time spent in the different areas to try to get a readout or an index of how anxious the animals are. And so normally in the light dark box, wild type animals will spend about maybe 90 seconds or in the light. And when we compare that to the heterozygous and homozygous animals, we see a significant reduction in the amount of time that both heterozygous and homozygous animals want to spend in this brightly lit compartment. And similarly, in the elevated plus maze, we see wild type animals normally have about maybe like a 25-75 split in terms of time spent. They spend most of their time on the closed arms and about 25% of their time in the open arms. And when we compare this to heterozygous and homozygous animals, we can see that both groups are spending significantly less time in the open arms and preferring to spend their time in the closed arms of the maze. We also were interested in what influence this loss of AIS synapses would have on oscillatory activity in the adult animals. Again, that was one of our original ideas, is that if we impair GABAergic signaling onto the axon initial segment, we might impair the patterning of activity. And so we did EEG analysis in the Gabra2-1 mice, and we use a Pinnacle system and implant animals with a small head mount, and then attach them to a tethered recording system. And this is just kind of an example of the types of recordings that we get from wild type and heterozygous animals. And we commonly analyze these complex waves using fast Fourier transform and also spectral analysis. So we divide them into different frequency bands, delta, theta, alpha, and gamma in particular, and then have a look at how these bands differ across the different genotypes. And again, we know that the slower oscillations like delta underlie things like sleep, whereas these fast oscillations underlie things more like working memory or active engagement in a task. And so down here, we're looking at a comparison between wild type and heterozygous animals. And this is a spectrogram plotting time, frequency, as well as power. And power is represented by the intensity of the color. And what you can see is down here in this low frequency portion, heterozygous animals have much higher power. And we can see that represented in the fast Fourier transform over here as well. So wild type animals are here, and heterozygous animals have much higher power, especially in this kind of low frequency delta band. And here's looking at average delta power between wild type and heterozygous animals. You can see that significantly increased. So in collaboration with AstraZeneca, we were also able to test some novel compounds to see if we could normalize some of the consequences of AIS synapse dysfunction. And so we had a look at these novel selective benzodiazepines. And these are benzodiazepine compounds, so they bind to the benzodiazepine site, but they're specific either for alpha-2 or alpha-1-3. And so we tested a compound called AZD7325 that's an alpha-2 selective benzodiazepine-like drug, or positive allosteric modulator. And again, here we're looking at the elevated plus maze paradigm. And what we can see is that we have vehicle-treated, diazepam-treated, and AZD-treated animals here. And we're comparing wild type in white to heterozygous animals in gray. And again, replicating our prior data, we can see that heterozygous animals treated with vehicle are much more anxious than their wild type counterparts. And interestingly, when we treat with diazepam, we can see a decrease in anxiety in the wild type mice, but the anxiety in the Gabra2-1 mice didn't seem to change. However, both groups showed a decrease in anxiety when treated with the AZD compound, AZD7325. And here in this graph, we're just looking at heterozygous animals and comparing the three groups in terms of their percent closed time. And we can see vehicle and diazepam-treated heterozygous animals are spending, close to 90% of their time, 90% of their time in the closed arms. And in comparison, when we treat them with AZD7325, we see a significant reduction. We also pursued testing some of these selective benzodiazepines for normalizing the EEG as well. And so here again, we have baseline recordings from wild type and heterozygous mice. And the red line here represents our administration of AZD7325. And so what we can see is that shortly after administration of the AZD compound, we see this powerful reduction in delta activity that's relatively long-lasting. And even though it still has a little more power overall in this example in comparison to wild type, this was a significant reduction in delta power. We can see that plotted over here. So this is fast Fourier transform analysis again. We're looking at normalized power and frequency on this axis. So this is the delta frequencies. And up top here, we can see these are heterozygous animals at baseline. And when treated with the AZD compound, they move down and largely overlap with the wild type frequencies. And here we're looking at theta frequency because we can also see some alterations in theta as well with the AZD compound. And so here we've got heterozygous baseline, and then we can see that suppression of theta by the AZD compound. So to summarize some of the research that we've talked about so far, the GABA-A receptor alpha-2 subunit we've shown is a preferential interacting partner with collybistin. And this is, again, kind of a new finding because we thought that Geoffrin was a partner for all of the alpha 1 through 3 subunits. So once we were able to identify that collybistin was a unique interacting partner of this portion of alpha-2, then we decided to mutate it. And so mutation of amino acids 358 through 375 of the alpha-2 subunit of the GABA-A receptor results in increased expression of the alpha-2 subunit, but it is mislocalized and it doesn't reach the axon initial segment. And we also see a corresponding loss of inhibitory synapses there. And so this suggests that even though other subunits can get to the axon initial segment, they're not sufficient to help wire it up properly from the chandelier cells. So when alpha-2 can't get there, we end up losing those inhibitory synapses onto the AIS. We also saw that a proportion of the Gabra2-1 pups die during this late postnatal period. And it was really in the range between postnatal day 10 to 25 with a peak right around postnatal day 20. And this is likely related to the seizures that we observed, although we were never able to kind of directly correlate an animal having a seizure and then seeing it die from those seizures. And this is an area where we're kind of concentrating a lot of effort now is to try to examine, what's happening during development. Are all of the mice having seizures and only some of them die? Or just a portion of them are having seizures? And also trying to kind of examine what some of the factors might be in whether animals die or survive during this sensitive period. We also found that Gabra2-1 mice also exhibit anxiety and impairments in the control of oscillatory activity in adulthood. And this was evidenced by this huge increase in delta power in the electroencephalogram. And then we also went on to test some novel compounds from AstraZeneca, again, to see if they could normalize the anxiety and also the altered EEG activity. And we were able to see efficacy of an alpha-2 selective positive allosteric modulator called AZD7325. So just to kind of bring this back around to human health and neurodevelopmental disorders, there's a population of human patients with mutations in collybistin, and they display a severe developmental syndrome that's characterized by sensory hyperarousal, seizures, hyperactivity, disturbed sleep-wake rhythm, anxiety, cognitive, verbal, and social impairments, and early mortality. And in this talk, I showed you that the Gabra2-1 mice have seizures, and they have enhanced anxiety, and they also have early mortality. And in other studies, we're evaluating their sleep-wake rhythm and looking at their levels of activity overall, as well as some of these other domains. And again, what we've seen so far is that the alpha-2/1 or GABA-2/1 mice really display common characteristics with these patients with mutations in collybistin. And so alpha-2 dysfunction may be responsible for these symptoms in this disorder and may represent a new therapeutic target. And also there are a number of related disorders that may be promising for treatment with alpha-2 modulators, such as Dravet syndrome, because again, they share a lot of these characteristics. So that's kind of, again, an expanding area that we're working on now, is looking at other disorders and do they share some of these features. So I just wanted to thank Dustin, who is my #1 collaborator, and we have a lot of really interesting projects that intersect between the two labs, so I feel really lucky to have him working with me. Stephen Moss, who was my postdoctoral mentor, where a lot of this work was started. Hans Merrick and Herman Schindlin, who contributed a lot of the biochemistry data analyzing the interaction of alpha-2 with collybistin. And Patrizia Panzanelli and Jean-Marc Fritchie, who were very helpful with some of the immunohistochemistry. And then Nick Brandon and Alan Cross from AstraZeneca, IMED, and Neuroscience, they contributed novel compounds for us to test in the animal models. And 2 current graduate students are contributing to these projects, and in particular in carrying them forward, Rochelle Ali Rodriguez and Austin Boren. And we have a number of really fantastic undergraduate students that work in the lab as well. And students that are working on this project most closely are April Contreras and Christina Hoya. And those are the slides that I have for today.
Good. Thank you so much. So if you can unshare the presentation.
There we go. Did it work?
Yeah, here you go.
Okay, great.
So thank you very much. It's pretty interesting. I hadn't known the details of like all these various GABA segments, what is happening and all those things, which is pretty interesting to know.
Yeah.
I have a few questions.
Sure.
You mentioned in the mutant there is a mislocalization, right? They don't go to AAS.
Yeah.
Have you observed they go to any other place other than AAS?
So there is more total receptor, and it does seem to be mostly diffuse. Actually, not mostly diffuse. There is a diffuse population of alpha-2 subunit. But when we look at co-localization of alpha-2 with presynaptic markers, we do still see that at other sites, alpha-2 goes to synapses. So we can still see co-localization of VGAT positive presynaptic terminals and alpha-2 on the soma, for example, and on the dendrites. Okay. So it seems relatively specific for the axon initial segment synapses.
So they might move to some other place to join.
Yeah, I mean, it does seem like they're at other types of synapses, and normally they are present at other types of synapses. It's just that they're preferentially enriched on the AIS. And there's also some ideas that there might be mixed populations at different types of synapses, and maybe even within a single receptor there might be a mixed population of alpha subunits.
Okay. And you had mentioned initially like the GABA receptors, like those go down with the age. Like initial first, second slide or so. And I have a question on that. Like sometimes like you see some of these epilepsy in humans, especially in the later part, like when they are grown-ups. So is it due to like, I mean, like, because I would think that development has already stopped in terms of brain development at that time. So is it because something else is happening in the humans?
So you mean in the older populations? Why do they? Yeah, so it's a bit complex when people get older. One thing that starts to happen, especially in the teenage years, is there's an increase in, head injury and things like that, and those things can predispose to development of seizures and epilepsy. And then later in life, there's a number of complex things that happen in terms of, just general dysfunction of circuitry. And when we look at GABA receptor subunit expression, alpha 2 is really high early in development and then goes down into adulthood, but other subunits start lower and then go higher. So it's kind of a complex pattern, and it doesn't necessarily map right onto epilepsy, especially in the older adults.
Older adults, okay. And in the neuron, like these GABA ones are like found in at different, like you mentioned that at different places, right?
Yeah.
So has anybody else has looked into what happens if you eliminate at other places?
So there's some relatively new work looking at what specifies the perisympathetic synapses. So Jean-Marc Ritchie's group has looked at a molecule called dystroglycan, and it seems to play a really important role for some of these synapses that are on the cell body.
Mm-hmm.
And so, they've looked at that mostly kind of on the cell and molecular basis, and we don't yet have too much information about what happens in those animals.
Okay. I think I'm exhausted with all the questions right now.
Okay.
So I really thank you so much for taking part in this hangout, and I really appreciate your support, and hope I'll be presenting this, putting it on the Facebook and our site, which you should be able to see within 24 hours on the main
Great.
page of our site. And as soon as we are done, you can see it on our web page also.
Great.
So
Thank you so much for inviting me to do this.
I really appreciate your support. Thank you.
Yeah. Take care.